Peace be upon you, ninth graders. In this activity, we will discuss the root of nutrients. So prepare your materials and let's get started. At the end of this video, you will be able to describe the structure of the inner wall of the small intestine, define absorption, draw and label a Velus, determine the fate of each nutrient after digestion, list the characteristics of intestinal wall that facilitate absorption. The cells of the human body are in constant need for oxygen and nutrients. Upon digestion, complex food molecules are transformed into nutrients. In the previous video, we have seen that nutrients are obtained at the level of the small intestine at the end of digestion. How do these nutrients reach the cells? What are the structures in the small intestine that favor their passage to their final destination? Before we answer these questions, let's recall our progress until now.
Can you believe your digestive system is as long as a bus? Yep, it's about 9 meters long, all wrapped up inside your body. A lot of things go on in your digestive system, not just digestion. What else do you think happens? Digestion goes hand in hand with absorption, which happens in your small intestine, which is what we're going to look at in this video. Digestion is the breakdown of food, from large insoluble molecules into small molecules, which can be used by your body. Digestion involves many organs before it reaches the small intestine. As they move through the digestive system, carbohydrates are broken down into sugars, proteins are broken down into amino acids, and fats are broken down into fatty acids and glycerol. But the job of the digestive system doesn't stop at digestion. For our body to make use of these small, soluble molecules, we need to absorb them into our bloodstream.
So, to get use of the nutrients that have been obtained by digestion, they must be absorbed into the bloodstream. Where and how does this absorption take place? Let's now study the structure of the small intestine where the nutrients are found. If we take a portion of the small intestine and we cut it open, we will see that the inner wall of the small intestine is not smooth. It is covered with circular folds or ridges, each of which is further covered with finger-like projections called villi. So, this is a portion of the small intestine cut open. You will see that the inner wall of the small intestine is not smooth. It is covered with circular folds or the ridges. The ridges in turn are covered with numerous finger-like projections called villi. Each villus itself is also covered with microvilli.
The structure of the villi is specially adapted for food absorption. The villi are finger-like and large in number. Each villus has a lymph vessel called lacteal in the center. The lacteal is surrounded by a network of capillaries.
How to draw an intestinal villus? First, you start by drawing a single layer of cells in the shape of a finger. Then, inside it, you draw a lymphatic vessel surrounded by a blood capillary.
Water-soluble food molecules such as monosaccharides, amino acids and minerals, as well as water-soluble vitamins, are absorbed through the walls of the villi into the capillaries. On the other hand, fatty acids and glycerol enter the epithelium of the villus by diffusion. In the epithelial cells, they recombine into fine lipid droplets. The lipid droplets then enter the lymph in the lacteal.
What is intestinal absorption? Intestinal absorption is the passage of nutrients from the small intestine to the blood or to the lymph. Then, the routes of absorption are blood and lymph. Glucose, as well as other monosaccharides like fructose and galactose, amino acids, vitamins and minerals all pass into the blood except for fatty acids and glycerol, which pass into the lymph. Absorption is mainly carried on by diffusion. But what is diffusion? Diffusion is the passage of a substance from a medium where its concentration is high into a medium where its concentration is low through a semi-permeable membrane. So in this figure, the concentration of sugar on this side is much greater than its concentration on the other side. Thus, by diffusion, the sugar molecules will pass from this medium through the permeable membrane into the second medium. The same concept applies to nutrients in the small intestine. The concentration of the nutrients in the small intestine is much greater than their concentration in blood and lymph. Then, by diffusion, the nutrients will pass from the small intestine into the blood or into the lymph. The intestinal wall is an exchange surface that's well adapted to its function. What are the characteristics of the intestinal wall that facilitate absorption? First of all, we have the large surface area. What do we mean by surface area, and why is it large in the small intestine? When we study the surface area in the small intestine, we mean the surface that is in contact with the nutrients. In other words, it is the surface that the nutrients have to pass in order to be absorbed. In order to understand the concept of the surface area, let's consider these two structures, A and B. After the folded structure B is stretched out, it seems that its surface area is much greater than that of A, the unfolded one. So, as the folding increases, the surface area increases. Does the folded structure B remind you of something? Of course, it is the structure of the small intestine. And don't forget, there are 3 levels of folding in the small intestine. First, we have the ridges, which are covered by villi, and the villi themselves are covered with microvilli. This will greatly increase the surface area of the small intestine. So, if the small intestine is stretched out like the folded structure B, its surface area will reach about 200 meters square, an area big enough to cover a huge house. The second characteristic is the thin wall. That is, the nutrients have to pass only one layer of cells in order to reach the blood or the lymph. And finally, don't forget that each villus is provided with its own network of blood capillaries, which will ensure the absorption of a great amount of nutrients. Now let's apply what we have learned. The following table shows the quantity of nutrients in blood before and after a meal. Analyze the results of the table. The quantity of glucose in blood before a meal was 0.8 to 1 gram per liter. It increased to 1.5 to 2 gram per liter after a meal. Similarly, the quantity of amino acids in blood before a meal was 0.4 gram per liter. It increased to 0.7 gram per liter after a meal. How can you explain such a result? The quantity of glucose and amino acids in blood increased after a meal since at the end of digestion, nutrients passed through the intestinal wall into the blood. Name the phenomenon that led to the increase of quantity of nutrients in blood. The phenomenon is intestinal absorption. Name the structures of the small intestine responsible for this phenomenon. The structures are the intestinal villi. Now, let's sum it up. After food is ingested, the digestive system will simplify the complex food molecules into nutrients that can be absorbed into the blood and transported to all body organs. Absorption occurs in the small intestine at the level of intestinal villi. There are 2 routes of absorption, the blood and the lymph. The large surface area, the thin wall, and the richness in blood capillaries are all characteristics that facilitate absorption. Always remember that all of the nutrients, including glucose and other simple sugars, amino acids, vitamins, and minerals, pass into the blood except for fatty acids and glycerol that pass into the lymph. And finally, you will be assigned to study paragraph 6, absorption, page 4 on the support guide. This is all, dear. Hope you enjoyed the lesson. Study well and keep safe.